really a reality. And of course, stem cell-based therapy is the ultimate in personalized medicine.
Recent research has eradicated the need for the use of embryonic or fetal stem cells for stem cell therapies. In fact, we can use the patient's own cells and implant them into the body and have the achieved therapeutic outcome.
Now that you've eliminated the moral and ethical question about acquiring embryonic stem cells, we've now unleashed on the world stem cells as the possibility of being coached to tell the human body how to correct itself.
Cellular therapy really in the last couple of years has just, the entire universe has changed.
I think for the first time we can really think about treating patients with therapies that will provide a permanent cure.
We now have patients that are long-term survivors from cellular-based therapies that might not have had a chance at even going into remission previously.
Prior to having the GMP facility across the street, there was really no facility like this within at least an 800-mile radius. With the location of Denver, you can even dream about potentially serving all of North and South America. Would you like to think big and dream big? I'm Dennis Roop. I'm the director of the Gates Center. I can honestly say that coming here has completely exceeded all of my expectations.
I'm Tom Payne. I'm Director of Cell Therapies here at the Gates Biomanufacturing Facility. I work directly with adult stem cells at this facility.
I'm Janelle Blessing. I'm on the advisory board for the Gates Center. I fell in love with the center and was blown away by the cures that they were coming up with. And I felt like I needed to be part of getting that message out, that this was happening on the Anschutz Medical Campus.
My name is Naresh Mandava. I'm the chair of ophthalmology at the University of Colorado School of Medicine. As a retinal specialist and a specialist in macular degeneration, we are really excited about our collaboration with the Gates Regenerative Medicine Center and are looking forward to all of the exciting technologies we can build together to combat these horrific diseases.
My name's Dick Jossi. I have macular degeneration and I'm a patient of Dr. Mandava's. That's why I'm so excited about when I hear all this. I want everybody to have that same advantage that I've had.
Honestly, I would never have dreamed that we would be where we are at the present time, but with respect to the research efforts that are ongoing.
I mean, I was literally listening to NPR and they were talking about stem cell biology and how it was going to change the face of science in 5 years, and then the next thing you know, I'm on a tour at the Gates Center and realizing that it's already changed there.
We are in the gowning room to go into the cell therapy manufacturing suites. We've designed the space to have clean and dirty sides. Once you come over this line, you have to be fully gowned from head to toe. So once you're gowned with a hairnet, tie-back suit, booties, you can enter into the processing area. So right now we're in the cell processing suite. This is where we'll do manipulation of cells and we'll isolate the stem cells. We'll grow up the cells in here. The incubators mimic the temperature and the physiological conditions of the body. The cells feel like they're in the body and they'll grow. There's less than 100 particles per cubic foot within this space here, so it's ultra clean space.
This is a golden era of personalized medicine. We can take your own cells. This amazing team will process those cells and convert them into retinal cells. I've received them in the operating room on that day, implant those cells directly into the macula. The prospect of doing that is phenomenal to me.
I would like to see the light at the end of the tunnel. But if I were to have that next step in my future, I'd be pretty excited about it. Not only for me, but for anybody else, in the future.
So what's so exciting to us about this facility and its potential is that not only are we going to be able to help folks like Mr. Jossi who have macular degeneration, but there are applications to virtually every area of medicine. We are also developing therapies and things like Parkinson's disease. We're looking for new ways to grow bone and cartilage. We are on the cusp of manufacturing real human blood. But maybe one of the most exciting areas that's under development right now is Dr. Roop's area of specialties in dermatology.
Adam is a remarkable little boy that I met first in 2006. We had actually discovered the genetic basis of his disease.
My name's Adam, and I'm 13 years old, and I have a skin condition called EHK, which stands for epidermolytic hyperkeratosis, and it thickens my hands and the bottoms of my feet.
They really lead a life of extreme pain from constant blistering.
It sort of restricts me from like playing sports, but now that I've like grown older, I'm able to like play like tennis or soccer with my friends.
So Alan, if you remember when you were here before, that was in the summer of 2012, we actually showed you under the microscope your cells that we were growing in the lab. But even though we were growing them there, we could never actually use those cells to treat you because we weren't growing them in a really clean environment. Now we have this great facility, so we can actually, at some point in the next few months, we'll have to take another biopsy of your skin, and then we'll begin to grow your cells in this room. Finally, then we have the GMP facility across the street. This really becomes a reality. So for me, it's extremely exciting. And we hope that in the very near future, that Adam will actually be one of the first patients that we actually treat using the new GMP facility. So that gives you a little bit of context for what we're really hope to do with this facility. And so what I'd like to do is just walk you through a little bit more background and emphasize from the very beginning that, you know, when I first came here, we made a decision that we would focus on adult stem cells and not embryonic stem cells. And there were several reasons for that. One, you may remember in 2006 and 2007, the Bush administration actually had a ban on using federal funds to actually create new human embryonic stem cell lines. And if you wanted to do that type of research, you actually would have to have a separate building. You couldn't do it in the same building where you had federal funding for other research. So pragmatically, we really didn't have enough funds to have a separate building. But so it was actually very fortuitous. I came up for a press conference in August of 2006, and just a few weeks before, a scientist called Semyo Yamanaka had made an amazing discovery that he could actually reprogram adult somatic skin cells all the way back to an embryonic-like state. So that was an amazing discovery, and many of us, I must admit at the time, thought, it's impossible. But in fact, it really turned out to be an amazing technology that was very robust. So within a year, Yamanaka and 4 other independent groups showed that you could actually do that for human cells. So basically, just to briefly introduce what reprogramming is, if you think about early embryonic development, the reason that embryonic stem cells were so exciting to investigators is the fact that they're pluripotent, which means that they can give rise to every cell type within the body. So very early, those cells, as we say, can give rise to every cell type, but as they begin to specify or differentiate into different adult lineages like bone or muscle or skin, you don't want a new bone growing out of the front of your forehead, right? So how does the body control that? And the way it's controlled is as those cells begin to specify, the stem cells that give rise to bone or muscle or skin, the genetic information that's in the nucleus is basically shut down and suppressed, except for the information you need to make another bone cell or another skin cell. And so basically what reprogramming does is it takes an adult cell that can only give rise to skin, let's say, And it kind of strips the memory of the nucleus where the genetic information is, all the way back to a ground zero state. So now those cells can actually give rise to every tissue. So if you think about it, not only does that get around the ethical and political objection to potentially destroying an early fertilized human embryo, it's actually better medicine because you actually take the biopsy from the patient that you want to treat. So we don't think we'll have to worry about immune rejection using this type of therapy. So, so that's just a brief introduction to reprogramming and the fact that we're either reprogramming adult cells, or in some cases we're actually isolating and either modifying or expanding existing adult cells from adult stem cells, and we're not using embryonic stem cells. So, this was all made possible by Charlie Gates. So, when I had been approached by the university about moving here, actually initially in 2001, they brought me out to this campus. There was one new building, and I said, "You gotta be kidding me. You expect me to leave the Texas Medical Center for Army barracks?" And every year they would invite me back, and by 2006 it was clear they were going to build this campus, which if you haven't had a chance to look around, it's an amazing campus. No other medical school in the U.S. has built an entirely new medical campus and research facility. So it really is an outstanding infrastructure. But the initial gift that helped the School of Medicine move us from, I was at Baylor College of Medicine in Houston, was a gift from the Gates family. So this was Charlie Gates, who many of you know. This was Gates Rubber, a local company. And Charlie was actually a patient at University Hospital over on 9th in Colorado. And shortly before his death, he was talking with clinicians. And being an engineer, I think Charlie was just intrigued by the idea that someday you could actually use stem cells to regenerate new tissues, maybe even new organs. So he convinced his children, this is Diane Gates Wallach and John Gates, that they should support stem cell research. So that's where the initial gift came from. And it was really Charlie's vision that he explained to his children that enabled all of this to start on this campus. And this family has been unbelievably supportive ever since. So this is just a schematic that we kind of use to explain how we got to where we are. So initially, there was a gift from, as I said, the Gates Frontiers Fund, $6.5 million, that allowed us to move from Houston. And then since then, the Gates Frontiers Fund has actually partnered with the CU Foundation. Initially, there was a $5 million gift that was made when we achieved center status in 2010. That was matched by the CU Foundation. That ended at the end of this past year, and both the Gates Frontiers Fund and the CU Foundation agreed to increase that to $1.2 million each per year. So basically, we have $12 million then to combine to run the facility for the next 5 years. So it's just an amazing example of one gift being matched by the university and its foundation to enable us to grow. So we've actually then leveraged those funds into now about 130 million in support, mainly from the National Institutes of Health, some from the Department of Defense, the Veterans Administration, or smaller foundations. With respect to philanthropy, we've now raised about 40 million. And this facility itself is again a great example of a partnership. And Tim will tell you about that in a few minutes. But we raised a total of about 16 million to build this facility. And Tim will give you the specifics. And then also commercialization. So we are a consortium center. So we have 82 members that are made up of faculty on this campus, as well as some from the Boulder campus, some from the vet school at Colorado State. We also have bioengineers from the Colorado School of Mines. So we basically really opened up the center for all scientists in the region or clinicians that are interested in stem cells and eventually applying those to some clinical application. I won't talk about the course facilities, but I would like for Tim to come up and talk about how we're actually trying to commercialize some of the technology that's being developed by our center members.
Sure. My name is Tim Gardner. I'm the Chief Financial Officer. I've been here for a few years, and prior to that, I raised money for high-tech companies with VC companies. So I kind of came from the commercial side of the world and have joined this incredible place where all these amazing things are going on. The mission, it's really driven by the Gates family and Diane Wallach, is really on commercialization. So she's interested in the raw science that's happening here, but she demands that it actually gets to a level of commercialization. So that's an area where we're a little different than lots of centers at universities, academic universities around the country, and we really focus on it. So the things about commercialization are we provide cutting edge equipment. The Gates Center itself, with a lot of that funding that Dennis talked about, our researchers, whether they're from CSU or here on campus or whatever, they need the newest, best state-of-the-art equipment to be at the cutting edge to continue that commercialization path, and that's kind of part of our core mission. The other thing is they need to document their intellectual property as early on in the process as they can, and then protect it, because that's really the path to fundraising and the path to commercialization. So we have our own PhD in biology and IP attorney who can really work with investigators very early on. And Children's Hospital Les was talking about is doing some similar things, but it's in support of commercialization. And then I get involved with the investigators very early on in the process to kind of talk about, is this a licensing opportunity? Is this really a business opportunity? If it is a business opportunity, who needs to be part of the team? We actually work with our investigators to go to the CU Technology Transfer Office and negotiate their first license and then raise money. So that's kind of a big part of the commercialization support, and it's baked into the DNA of the Gates Center. And when we get to the next level of it, this facility is a key step in getting things out of the lab and into commercialized therapies. So that's kind of the commercialization message. So as Dennis talked about, we have this consortium of 82 members. We work with other universities. I talked about how our core labs, but the next key thing is, and this, we talked about the incredible nature of this campus, that 15 years ago there was Army barracks here, and now there's 2 world-class hospitals, the University of Colorado School of Medicine, which is in an incredible growth phase with the amount of people that we can attract that are coming here. And so that's really access to patients. So you kind of can complete the loop. The loop is you invent it in a lab, you get the process improved in a facility like this, then you get the therapy approved for an initial phase 1 clinical trial. That happens at the hospitals. You maybe fine-tune it, then you do a multi-site phase 2, and then all of a sudden it becomes a therapy. And that can all happen now at Anshin's medical campus. And the Gates Center has had kind of a driving role in some of the elements of that.
So I'll just come back and make a point about Diane being very business-minded. And so when we first approached the school about building this facility, Don Elliman, who is our chancellor, insisted that we put a business plan together. And of course, to a scientist like me, a business plan is something you scribble on the back of an envelope. And I learned very quickly that I think Don's words when I sent him something was, "You gotta be kidding me." And so that's what I learned. We really needed help from individuals like Tim. We brought in consultants to help us with the business plan. But Diane insisted, and I think this is just something that's great about her leadership, that we have partners. And she said, you know, from my end, the money is the easy thing. The difficult thing is the politics, and that's where you've got to get everyone to play together on this. So, you know, we went begging. Of course, we had the business plan, and I think, you know, when you present a plan to CEOs of hospitals, they can really show them that, for example, with Children's Hospital, they're in the top 10 in the country. I think maybe 5, they're ranked 5 now. They were the only hospital in the top 10 in the country that did not have access to a facility like this. So to a CEO, they understand what this means with respect to recruiting new faculty with cutting edge technologies and then bringing that to their patients. So we were able to get both Children's Hospital, University Hospital, and the School of Medicine to partner with us and helping build this facility. So again, that was Diane driving. She always wants to leverage her money as much as possible by other people, in essence, having skin in the game. So I'll just briefly go through these. I mean, these are just examples of technologies that are currently in development on this campus, and in some cases there have actually already been startup companies that have been spun out. They're actually in this building next, the older part of this building. They're in incubator space developing new startup companies. So the first is a universal blood supply. We have a young investigator, Joseph Ripelli, who has developed a way to grow and expand human hematopoietic stem cells, or human blood stem cells. And so if you think about it, one of the things that you can do with those cells is once you have a human blood stem cell line that you know is pathogen-free, you don't then have to worry about getting hepatitis or HIV from a unit of blood from the Red Cross. And so Joseph is actually growing and making human blood in the laboratory from these blood stem cells. And so you can imagine this blood has a shelf life 5 times longer because it's made in the laboratory. It's all synchronized. They have the same age. So the shelf life is 5 times longer than an ordinary unit of blood. So you can imagine the Department of Defense is very interested in a blood supply that you can ship to remote parts of the world. And in fact, the DOD has actually funded that project. Restoring vision, you saw Naresh Mandava, who is a retinal specialist in the video. So the good news is another great partnership. The Gates Frontiers Fund put up a $5 million match, and just before the end of the year, we were able to find other individuals in the community to match that $5 million. So we had $10 million to develop a new ocular stem cell program, which will focus on macular degeneration. We helped recruit a young scientist from the University of Texas Southwestern who is working using, we call these reprogrammed cells induced pluripotent stem cells, or iPS cells, because we've induced them to a pluripotent state. So he can actually differentiate those cells into cardiomyocytes. And as Bob, we were talking earlier, one of the first times he came to the lab, he saw an example of this. And when you differentiate these stem cells into cardiomyocytes, they actually beat together and the culture of this, so you can actually see them beating through the microscope. I'll come back in here to skin diseases. That's what I worked on. We recruited Karen Payne from University of Pittsburgh to develop a new orthopedic stem cell program. So she's working on differentiating these induced pluripotent stem cells into cartilage and bone lineages, with the idea of repairing joints and for spinal fusions. I won't talk about cancer since it's a little bit complicated. We do have investigators working on neurodegenerative diseases such as Parkinson's. If we have time out in the hall, I can show you an example from Kurt Fried, who's a pioneer, where he has differentiated iPS cells into dopamine-secreting neurons. This is the neuronal type that's destroyed in Parkinson's patients. So this is a potential example of how this technology could be used for those types of disorders. Let's go on to the next slide. So just to give you an example of work from my own lab, so basically the idea is we take a biopsy from the skin from one of these patients who have these inherited skin blistering diseases like Adam. We put the cells in culture, so these are just their skin cells that still have actually the genetic defect. We put them in culture, and then we've developed a way to reprogram these cells into these induced pluripotent stem cells that's non-genetic, so we don't use viral vectors. So in thinking about getting something approved by the FDA, this is a much safer technology than Yamanaka originally developed back in 2006. So we've actually filed a provisional patent for that technology. So once you have these cells, they're just like embryonic stem cells in that you can grow them indefinitely. You can actually freeze them and store them for years. You can thaw them, they're still viable. But since you can grow them and make a lot of the cells, we can actually think about correcting the genetic defect that causes the disease. So if you think about a disease like Adam's, one way to think about the human genome is to think about the largest book you've ever read that has 3 billion letters. So that's basically how we think about the genetic code in letters. There is one typo in one letter that causes Adam's disease, just a single typo. But now that we know the entire sequence of the human genome, we can look for sequences around where that single typo is. We can develop a new series of technologies called gene editing technology to basically engineer little molecular scissors that will bind and cut the DNA very close to where the mutation is, and then we insert in the normal sequence. So that's all doable. We do it routinely in the laboratory now. So once you do that in these pluripotent stem cells, we select out the cells that we corrected, and then we can actually differentiate them back into adult skin stem cells and return them back to the same patient as an autograft. So that's just an example of how this technology can be used, especially in diseases that are what we call monogenic or single gene defects, where we know exactly where the defect is. So one of the great things about this campus is the location. So basically, with our patients, just a 5-minute walk away in either Children's or University Hospital, we can go and remove either tissue or maybe draw blood that contains immune cells, lymphocytes. Just walk those cells over to this facility. We have some amazing new pieces of equipment where you can basically take a peripheral blood, a unit of blood drawn from a patient that has lymphocytes in it. It's a closed system. This system will actually pull out the lymphocytes. These are the cells that fight either infection, or in some case you can actually modify them to make them attack cancer cells. So this is, we're currently involved with a recruitment. I'll come back to that with Children's, with Leo Gore, of trying to recruit an individual that's a world leader in this technology that would use this facility. So basically you would take peripheral blood from a patient across the street, modify their lymphocytes, and then expand them and deliver them back to the patient. And there have been some amazing complete cures of different types of cancer, especially leukemias, during the last 2 years with this technology. So again, these cells would be isolated, expanded. We would package them in some way, put them in a little container that we would seal when it leaves our facility, walk them across the street. It would be opened in one of the procedure rooms where those cells would then be returned to patients. So again, the great location of being just across the street, you minimize a lot of risk of having to transport cells either across town or even out of the city, just by having the patients right next door. So this is just what these facilities look like. We'll walk you, we'll take you on a tour in a few minutes. So we have, GMP stands for Good Manufacturing Practices. So this is just, Tim can explain this better than me, but it's a combination of having a very clean facility. So our air is filtered. There are very few particles in the air where we actually grow and expand the cells, and we'll show you what those areas look like. And we also have to have a process that's very tightly controlled, unbelievable regulations that the Food and Drug Administration requires that we have in place, so they know that we can reproducibly grow a cell type, and we can know by its properties that it's going to behave a certain way when we return it to a patient. So all of that has to be documented and verified. So that's basically what GMP stands for. So we can do both cell processing in a facility like this, or we'll show you in real life what this looks like, but that's a large fermenter where we can actually grow and expand the production of proteins for biologics. So we have some faculty that have examples of a biologic that could be added directly to tissue to actually accelerate the repair process. So we have that capacity as well. And we already talked about the partnership. So, Tim, do you want to talk about quality management?
Yeah.
We have a guy who actually...
He thinks in this way. This is what he thinks about it. If you ask him a question, he thinks in flowcharts and stuff. And he's our quality assurance guy, which is just where you want him to be. Just to back up a little bit, so when we talked about sort of, Dennis said, you know, we had an idea on a napkin and created a business plan. There was really two objectives that we had there. One is the University of Colorado and the campus, both hospitals, wanted to be able to recruit both nationally and internationally the absolute best researchers and clinicians in the world. And in a number of cases, they came to the facility, they liked the team, they liked the opportunity, but they said, well, where am I going to be able to create GMP grade materials? And when the answer was, well, we're thinking about building one, they said, well, call me when you grow up. And they did not continue the recruitment, and we lost some great people. So one of the things that we wanted to be able to do is to stop that from happening. And actually, we haven't locked in any yet, Dennis will talk about it, but we have two people that are of that world-class caliber that, but for this facility, would not be talking to us. Now, it's necessary but not sufficient, right? It's one of the pieces, it's not the whole pie, but we have actually done that. The other hope was that with biotechnology companies, with intellectual property that was invented in Colorado at the Anschutz Medical Campus, we wanted to keep those people here. And we have one customer that was searching for CMO facilities worldwide and has now come back and is working with us. And more importantly, a company that is based in San Diego, that's a public company with $300 million in cash, just rented some office space just down the hall from us, and we're likely to be doing ongoing service contracts for them. So from a business perspective, the idea of build it and they will come, which is a difficult sell, particularly if you're trying to raise money around that concept, it's actually working. So that's really what we have going on here. Just to elaborate a little bit on what Dennis said, so GMP to me as a business guy means that we have an environment that is absolutely controlled, and then we have a quality management system that drives everything through that facility by a standard operating procedure. And so if you go and look at a piece of equipment, there's a standard operating procedure about how to turn it on, about how to calibrate it, and it has to be certified to be within calibration at each and every time that you do a process on it. The people who are using it have to be trained in the use of that piece of equipment. And it kind of goes on and on and on. Any piece of material that comes into this facility has to come from a GMP grade facility. And if we have any question about it, we actually fly out and audit them. And then when the process occurs in this facility, everybody has a standard operating procedure. They're all trained to do it. We monitor the environmental conditions that were occurring as of the time that happened, and that's both particulate matter in the air, the temperature, the quality of the water that was used, all of the gases that come in and out of here. And then we go right into an analytical lab where we analyze every little piece of that. So as you can imagine, there's a lot of overhead and a lot of complexity to this, and that's why a lab on a campus can't do it, but we can. So what we have up here is just a schematic of all of that, but it's qualified suppliers, qualified equipment, qualified personnel, standard procedures, and intellectual property, and ultra-clean facilities. And we'll show later, and maybe we can kind of go through here.
Yeah. So you use Six Sigma or something like that?
Well, what we use would be Six Sigma, but it's actually the CGMP guidelines, which come from, basically, you have to adhere to the FDA Code of Federal Regulations about the individual materials that you're shipping out of here for human clinical trials. And in order to do that, there are these good manufacturing practices that you are expected to use. So I would say Six Sigma is light compared to the level of quality systems that we have in place here. So just to talk, you were talking about how special this facility is. There's maybe 24 to 30 academic GMP facilities in the country. There's other facilities like this, but normally when you build a facility and invest millions of dollars in it, it's usually dedicated, the whole facility is dedicated for one specific biologic drug. And they run it until they're finished the cycle on that drug, and then the facility's closed down or sold or repurposed. The academic facilities, we can help do primary research, and we can be a bit more flexible, and we limit ourselves not to going into final production, but into phase 1 and 2 clinical trials. Lots of the academic facilities, we did a lot of research. It was when we first kind of started to open, understand sort of what our positioning was. About half of these on this chart are the result of one particular researcher that had one particular product or therapy that they wanted to kind of create GMP grade material in-house. The systems aren't necessarily robust, the environment isn't necessarily robust, and an outside commercial client would probably not consider them as a potential contract manufacturing organization. Then the other half invested a lot of money like we have and can compete both for commercial opportunities and for internal research opportunities. So the facility you look at here is really at the top of the GMP facilities in America. And we know that because we've had 4 or 5 different commercial companies proposing to spend millions of dollars with us, send their quality gurus in here, and they've left saying, yes, this is a great facility and you ought to continue the negotiation. So it's not us bragging about it, it's actually potential clients really kicking the tires. So that's what the ground looks like in the United States for all of these facilities. But if you look at ones that are able to do both protein and cutting-edge cell therapies, there's really a very small group, and as Dennis said, we're the only one within about 800 miles. And just one other thing on the on kind of the quality side of cell therapy, there are not lots of FDA rules, and the FDA doesn't even understand about these cell therapies because we're in the early stages of even doing the first clinical trials in the United States. So the FDA relies on academic GMP facilies to help them learn how to regulate us. And they like academics because we're not greedy, profit-motivated commercial entities. We are academic and research-oriented, and our mission is really to accelerate research into clinical trials. It's not to make money for shareholders. So we're actually engaged, our employees here, in helping the FDA figure out how to regulate us and everybody else as we go down the path of new cell therapies. So here's more on the research.
Okay. So these are just some things that, different projects that are ongoing. So I didn't talk about cancer, but this is a project that's directed by Dr. Antonio Romino. He's an outstanding medical oncologist. And it's basically we now know that cancers themselves actually do have a stem cell population. And in fact, most cancers arise from the adult stem cell that normally replenishes and rejuvenates that tissue. And so now that we know these cells exist, you can actually try to identify a new class of drugs that actually target the cancer stem cells. So if you think about radiation therapy, chemotherapy, those therapies are directed against rapidly dividing cells. And in many cases they actually work, but we all have examples of friends or family members that 5 years after chemo or a radiation therapy, they relapsed and the tumor came back. So we think the reason the tumor comes back is the cancer stem cells are actually resistant to those therapies. And so now that we know these cells exist and their properties, Dr. Jimeno is actually running clinical trials of new drugs that just target the cancer stem cells. So it's much more focused. You eliminate the side effects like hair loss, GI tract disturbance, destruction of blood stem cells, and patients becoming anemic, because you can target the cancer stem cell and not normal stem cells. So very early stages, but we're very optimistic about finding new drugs that will be much better.
Can I comment on this real
Sure.
quickly? There's a heavy emphasis on commercialization, and I think this is a really excellent example of why. And I just want to make sure to sort of, I'm kind of the touchy-feely part of the ledger. Let's just say, for the sake of argument, that this drug that Dr. Jimeno is testing becomes New Drug Inc. or New Drug Company and yields a tremendous profit. The reason that the Gates family is so focused on us making sure that we protect that and develop it is that so the revenues from such a drug or a therapy can come back to funnel research. I heard a gasp when we presented the slide that showed $22 million of operating. I assure you, I drive an 09 Tacoma that's really beat up, and I have a threadbare shirt. What we do with 100% philanthropic money, 100% zero state money comes into our operating dollars. So what we do with our operating support, which is all philanthropic, is we invest it in opportunities like this. This opportunity came up within the first 5 years of the Gates Center's existence. And Dr. Jimeno was stuck because the National Cancer Institute, which is a part of the National Institutes of Health, has developed this irrational adherence to proof of concept, which flies in the face of its very existence. The NIH was formed to take high risk, and they now say, great idea, too risky, come back when you've got it proven. What Dr. Jimeno has done, and I see Leslie shaking his head because he understands this, is he has said, Dr. Roop, if I can get a small sliver, and in this case it was $250,000, a small sliver of philanthropy to invest in a proof of concept, I believe I can convince the NIH and the NCI, National Cancer Institute, that we have a winner. And in fact, he did. So this was actually a trial that was able to save a stage 4 cancer patient from a death sentence. And there are now 13 trials underway because a sliver of philanthropy, yes, this is a commercial, a sliver of philanthropy was able to be, but it's important because it's part of that 22 million that you saw operate, that we operate with over a 10-year period. We insert that at exactly the right moment to accelerate these and sort of poke at that National Institute gold basket so some of that money starts to trickle down. And that's how this happens. And again, what Diane is saying, great, save lives, change the world. And when you do so, protect that intellectual property so that in the future, we don't have to have commercials at events like this. We can show that we have sort of a self-sustaining enterprise, which is really what we're trying to build here.
So speaking of that, do you then share the technology so that other universities and research facilities can build upon them? Or do you have to closely protect it to keep the money and knowledge here?
The answer is yes to all the above, and I'll just, it's a complicated answer, and I don't mean to be dismissive.
So Patrick, let me just
Actually, sure.
Well, one example that we're just doing now. So even with these rare skin diseases, there is a new foundation. It's called the EB Research Partnership that's directed by an amazing young father who has a son with EB, who is a Wall Street guy. So we are just in the final stages of signing off an agreement to enter a consortium with Stanford and with Columbia University to take our technology, share it with the other. It's protected, but we have everything in place to protect each institution so we can more quickly accelerate getting this new therapy into patients at different locations. So that's just an example. I mean, you do have to protect the IP, but there are ways that you can structure it so you can share at a very early stage with other institutions.
I do have a question. And so, to stay with your example, so you got the therapy to find the typo, the one letter, the 3 billion.
Right.
And so is that the theory also that once you corrected the typo, that you will prevent the body from auto-correcting back to the wrong genome, to the wrong cell, and not bring the disease back?
That's a difficult question. I mean, you know, we don't think, you know, with these inherited diseases, they're really mistakes of nature that occur very early in development. And so it's unlikely that once we correct the defect and return it to a patient, that correction would be, we think it will be permanent. It could be reversed, but you know, you could say, why focus on the skin? Why focus on a rare disease? Our strategy, because no one in the U.S. is approved for this technology, we think it may be one of the first. One, the skin is accessible, so we can monitor it. It's not an internal organ. And there is no cure for these patients. Some of them will actually die very early in adulthood because they develop very aggressive skin cancers because they blister and wound repair so frequently. So we think it's a pretty compelling group of patients that we could convince the FDA, give us a chance to try it on these patients. If it works for these rare diseases, then we can expand it to more common diseases. So you kind of have to pick the lowest hanging fruit to initially try, because no one, the US is not approved, the FDA has not approved one of these iPS-based therapies. So it's very early stages.
I just have one follow-up on the intellectual property. Basically, when you protect your property, file a patent or whatever, a couple things happen. One is the research becomes publicly known, which is good that people can figure out that it's out there. Then what typically happens is people grant royalty-free research capabilities. So typically, if you own some intellectual property, you want as many people in the whole world using your intellectual property to do their research to try to build on it and have the impact. And if you have properly documented intellectual property, then when somebody is actually engaged in the commercial fulfillment of that, which is actually the objective, right? You also want as many people in the world using it for real-life therapies to help humans, then you at least have a seat at the table because you invented it. So it's actually not it's not intended to be restrictive, other than, yes, you want to be able to have control over who practices it and what the economic terms are. But the research licenses are freely granted, and everybody's on the same page. Does that make sense?
Yes,
Yeah.
thank you.
I've got a question more related to the commercialization side of it. So let's say this particular drug makes it big commercially in the market. How is the return coming back to the Gates Center? Through residual income in terms of stock? Or is it some sort of a royalty fees, or is that?
So there's two things. If you're doing research on this campus and employed by the university, and it's also true of the hospitals, any intellectual property developed is owned by the institution. So if a license occurs to either an individual company or a big pharma, then there is license revenue that comes back, and that's split throughout the university. There's, the university takes some, the inventor takes some, and then the department that the university worked in takes some. So that's an area where, with no other investment, there would be a return, and the university has examples of, multi-million-dollar license fees coming back to it for inventions, Botox and things like that. Then we are unique, and I'm glad you asked. We have a special program where it's called the Gates Grubstake Fund. Again, the Gates family had the wisdom to kind of think about this. And in cases where a company is ready to commercialize something, And so they're now too far along for NIH funding, but not ready for VC funding. There's a gap there. And in those situations, we are basically entering, it might be, say, a $350,000 grant, and they might not have even formed a company yet, so there's really no securities to participate in. We would have a letter of understanding that would say, if you do a qualified round, we would just like $350,000 on it, on whatever terms it is, whenever it is, and we're not trying to get credit for the fact that we were the first people in and should get more because of all the risks we took. We're just kind of tag along, and whenever you get something, remember us. It would be not even enforceable, but it would be an understanding.
So there's never equity in the picture.
Well, there can be yes. So there are situations where a company has spun out their separate company. The chancellor of the University of Colorado is looking at a fund that would act exactly like a VC and would actually purchase those securities. And that's actually happening on this campus too. So I would say we're not at the front of the train like some institutions like Stanford and others have been doing this for years and years, but we are quickly gathering resources to take positions in these companies to get economic rewards and to kind of keep them in our biotech ecosphere and not have them go somewhere else.
Thank you.
Okay, let's just quickly run through the rest of these. I talked about Gil's Felly, his ability to expand hematopoietic stem cells. I won't get into the work on HIV and influenza. Allender is one of the first examples of again, technology of a member of the center. Tim was involved in helping this investigator spin out a company last summer. They've raised the first round, and this is actually a biologic. Fred may know patients who get radiation therapy often, or chemo, can develop something called oral mucositis, which is really very painful lesions in the oral cavity. And sometimes that will limit the amount of therapy that they can actually get. So Xiaojing Wang, who's this investigator, has actually discovered it's in preclinical models that she has a biologic that you can just topically apply before radiation therapy and completely block oral mucositis. There's currently not a drug on the market for this indication. So the next step is actually, this is why we have such great relationships with the vet school. The vets up there, dogs that get radiation therapy and chemo also develop oral mucositis. So the first large animal test will actually be done in collaboration with the veterinarians at CSU. And if those prove to be as effective as the small animal studies, then since that's a biologic or a protein, which is much more familiar with the FDA, that could very quickly gain approval for a clinical trial. So this is why we also built the protein manufacturing suite for developing biologics like this. I won't talk about this. I mentioned a few minutes ago, we have this technology that's non-genetic for reprogramming. We filed a provisional patent on that. Tim talked about attracting world-class talent. So we have 2 recruitments that are currently underway that are in advanced stages. One is an investigator at the National Cancer Institute. This is this technology of modifying patients' lymphocytes. This is a recruitment with Children's Hospital. The Adore is leading that recruitment. We're helping with it. That individual needs this facility in order to be able to perform those types of therapies on this campus. We have another recruitment from Hopkins. It's a very similar thing. This is a cancer vaccine. Again, that candidate would use this facility. And then finally, this is a recruitment that all just happened. There was a bioengineer who actually trained initially at Boulder. He's on the faculty at Rice University. Just like 2 weeks ago, he agreed to an offer. It's a combined appointment from our bioengineering department as well as pediatrics. An investigator called Jeff Jaycott. He has an amazing technology of actually for kids that are born with congenital heart defects, of actually taking cord blood from the mother, growing and expanding those cells into cardiomyocytes or heart muscle cells. He has a matrix. So currently, we have fetal surgeons, and will actually, kids that have fetal heart defects, they actually will perform heart surgery on the fetus to correct basically a hole in the heart tissue with a patch, but it's not a living patch. So as the kid grows, that patch has to be replaced over and over. So this idea would be a living patch that's made with cardiomyocytes that came from the mother's cord blood, and it's a bio matrix that supports its growth. So in this case, the living patch would be inserted with the idea that it would grow with the child. So he was just recruited, so we're very excited about that recruitment. So just another great example of collaboration between bioengineering, which is a relatively new department that just came on this campus 2 years ago. They have the new building just down the street with pediatrics and also cardiology. So I think I'll stop there because we're really pushing 1 o'clock, and I know